Hello again. In this video, we're going through an activity that's in the textbook on pages 686, 687 that looks like this, where we're going to be using glass beads to represent chromosomes. We're going to take a population and do some pretend generations of it where parents have offspring and then those offspring have offspring. And then at the end, we'll do some number crunch with the data we've collected, and we'll see if the Hardy-Weinberg relationships that we've been learning about actually work, or if we've been lying to you all this time. So, the way this is organized, each of these things is going to represent a chromosome, and we have two types. We have dark blue ones, which are going to represent dominant alleles like big A, and then we're going to have clear ones, which represent recessive alleles like little a. And I've got 48 of the dark ones and 32 of the light ones per the directions on page 686. And what they have us do with those to start off is we put half of them on this plate and half of them on this plate, and this plate represents a bunch of males in a population, and this plate represents a bunch of females. Or rather, this represents sperm cells, and this represents egg cells. And what's going to happen in the next slide is we're going to start taking one of the sperm cells, one of the egg cells, combining them, and that'll represent an individual from the next generation. So we do that once, and what we have here is a heterozygous kid because they got a dominant allele from one parent and a recessive from the other parent. And then we do another one. This time we get recessive twice, and we do a bunch more. And what I'm going to show you in a minute is the first generation that I used, where I did this 40 times. I took one sperm cell from over here, one egg from over here, and combined them, and we did that until we had 40 offspring. And as you're probably used to from doing the Hardy-Weinberg formula, we want to know how many of these individuals are big A, how many are big A, and how many are little a. So we're actually going to go through and count and get some numbers from that. Now, in the book, I wish they used notation like this, but instead they talk about the bead colors. So big A, they're going to call dark. Big A, they call dark light. And little a, they call light. So, same thing. So, here comes the counting. I'll try to get through it quick. For homozygous dominant, we have 1 there, and 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Heterozygous, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 again. And because we know there's 40 of these, 16 plus 16 is 32, there should be 8 of the recessives, and let's make sure that's actually right. I am totally capable of miscounting. 1, 2, 3, 4, 5, 6, 7, 8. Good. Okay, so that's our first generation, and because I have to flip the page and I don't want to lose these numbers, I have prepared a tracker sheet where we can keep all this stuff. So first generation, we had 16 of them, and 16 of them, and 8 of them, and 16 plus 8 makes 40. Okay, now in the directions they tell us to do this for 4 different generations. So what happened after this was I had all these offspring. They said to consider half of them female and half of them male. So I decided this top group here would be the females, and the bottom group here would be the males. And so I took those offspring and I dumped them back into the 2 plates. So here's our new group of males, our new group of females, and now we're going to pair them up again and produce a new generation with 40 new offspring. And there they are. So now we get to count again. They have us do this 4 times, so I'm going to go a little quiet here and just collect data. Follow along if you like, or you can just skip ahead to where I get my finished numbers. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 for the homozygous dominant. Hetero, we get 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. 20 heterozygous. 14 plus 20 is 34. There should be 6 of the recessive, or the homozygous recessive. Are there? 1, 2, 3, 4, 5, 6. Good. Those numbers check out. We will record them and move on. 14, 20, 6. Total is still 40. So we split those up into the male and female plates, and then we made them up to make another generation. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 again. And for hetero, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20 again, which means this last number has to be 6 again. So we just got 2 generations in a row that had precisely the same statistics. That's certainly not guaranteed to happen, but it totally can. And total still 40. And one more time. What have we got now? Homozygous dominant, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 this time. So generation 4 has 15 homozygous dominant. For hetero, we get 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. +15 plus 18 is 33, so there should be 7 homozygous recessives, but let's make sure. 1, 2, 3, 4, 5, 6, 7. Good. Okay, total of 40. And now a grand total for all those generations. Let's put that here. Total. 16 plus 14 is 30, 44, 59. Is that right? 30, 40? Yeah, 59. 36, 56, 74. And 14, 20, 27. And grand total for that is 160, of course. Okay, so that's everything from what they call Part 1, demonstrating genetic equilibrium. I'm going to do all the analysis together at the end. I hope that doesn't throw you off too badly. So for now, we're just going to park those numbers, hold on to them, and I'm going to jump over to the next section that they call Part 2, testing for genetic equilibrium. That's on page 687. It's pretty similar to this, but with one difference that's mighty important to the beads. And we'll see how different it is number-wise. So in the new one, we again set up with the same gene pool at the beginning. We still have 48 dominant alleles and 32 recessive alleles. The difference is now, when we pair these up, the recessive trait is lethal. So, oh, sorry, yellow on white, not a good idea. So if we have big A, that's going to be a normal bead creature, whatever these are. And if we have big A, that also will be normal, because normal is dominant. But little a will die. it's some trait that causes a deformed heart, or you can't process an amino acid, or something that just kills you so that you don't get a chance to reproduce. And we'll see what that does to the numbers as we go through generations here. So just like before, we lay out these numbers of individuals, and let's see what we get. In our first generation, homozygous dominant, we get 1. That doesn't show up worth a darn. Let's change it. We get 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Big A, big A. Then we get some heterozygous. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. 15 plus 18 is 33, so there should be 7 recessives, I think. 1, 2, 3, 4, 5, 6, 7. There are indeed 7 recessives. That's encouraging. So, we've got those numbers, and we will record them in a second. Actually, let's get that done now. So for the second run, generation 1 had 15 homodominant, 18 hetero, 7 homorecessive. That's still a total of 40 individuals. Now the new thing, little a means death. So that individual dies, that one dies, that one dies. 4, 5, 6, 7. Those never grow up. They never have kids. This is what our population looks like afterwards. I've taken away all the homozygous recessives. You can see the empty spaces where they used to be. This is the graveyard where I kept the beads that didn't make it. And this is our new population. So we're going into generation 2 down a significant number of beads. And notice the recessive allele is the only one we are losing. If you have two dark blues, you survive. If you have even 1 dark blue, you survive. The only way you die of this is if you're clear or light. So this population should shift, and we should see it looking more and more blue as this trait starts to die off. And we'll see how that looks as we crunch the numbers at the end. So leaving these in the graveyard because they didn't make it, I took the rest of the population and said everything in the top half is female. 2 of them died. And everything in the bottom half was male. There's 1, 2, 3, 4, 5 males who didn't make it. So that meant that these were a little bit uneven. And then I took these and started pairing them up to make new offspring. And here's what we got. So this is our generation 2. How do they look? Away we go with the counting. For big A, we get 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Big A came out to 18. Then we did the heterozygous. 1, 2, 3, 4, 5, 6, 7, 8, oops, missed one, 9, 10, 11, 12 heterozygous. And we don't have a total of 40 anymore, so I can't use that shortcut. We actually have to count 1 double recessive, 2 double recessive, 3 double recessive. Why do I keep going to yellow? It's not a good color for this. 3. Okay, so there's our data. We can record that. 18, 12, and 3 for generation 2. Uh, 18 plus 12 is 30, plus 3 is 33. So we had a population drop because of the lethal allele. And now it happens again. This individual dies. so does this one, and so does this one. So now they're gone. The graveyard gets a little bigger. And now we take these groups and we say, this is our new batch of females, this is our mothers, this is our new group of fathers, and we go through 2 more generations of this. So here is generation 3. I think you probably get the idea by now, so I'll just try to count a little faster. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. Heterozygous, we get 1, 2, 3, 4, 5. And homozygous recessive, we get 1, 2, 3. Oh, shoot, did I miss a heterozygous? I think I did. I apologize. I want to get these right. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. 21 was okay. Heterozygous, 1 there, 2, 3, 4, 5. Yeah, it was 6. I did miss one. And homozygous recessive, 1 there, and 2, still 2, 3. 21, 6, and 3 are the revised numbers. Generation 3 goes 21, 6, 3. That's a total of 30 individuals. So the double recessive ones die off. We lose them. They go to the graveyard. This is our new group. And again, I take the top half to be my new females, the bottom half to be my new males. And here's what we got for our final generation. So last count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21 for our homozygous dominant. For hetero, we get 1, 2, 3, 4, 5, 6. And in this generation, even though there are still clear beads, we didn't get a single heterozygous recessive. So this generation lucked out. But we still have these future mothers and these future fathers carrying the lethal allele, so it may pop up again. So a trait like this can appear that it's gone away. It probably hasn't gone away. It's probably just that there are undiscovered recessives that haven't bitten you yet. So here we have 21 and 6 and 0, and that's a total of 27 individuals. So this population declined kind of quickly at first and then slower later on as the lethal allele killed some of its carriers, or the offspring of its carriers. So what did we get for our totals for that? 15 plus 18 is 33, 54, 75. Heterozygous dominant. 18, 12, 6, 6 gives us 42. And 7330 adds up to 13. And our total, 40, 73, 103, 130. Okay, so there's all our data. And we're done counting beads. Now let's get into the analysis that they ask for on page 687, and we'll see what that can tell us. So I'm just going to snapshot this data so we can actually have it on a slide instead of having to flip back and forth all the time. Good. Get rid of the clutter around it. Okay, so let's do some number crunch. What I'm looking at now is on page 687. They say, use the class data from part 1 to graph the genotype frequencies over 4 generations. Compare results with your prediction. So for part 1, the genotype frequencies would be 16 out of 40, 16 out of 40, and 8 out of 40. In other words, what we're finding from Hardy-Weinberg is our p squared, our 2pq, and our q squared. So here we get 16 divided by 40, which is 0.4. 16 out of 40 is 0.4 again. 8 out of 40 is 0.2. So that's our first generation. In the next generation, it goes to 14 out of 40, which is 0.35. 20 out of 40 is 0.5. 6 out of 40 is 0.15. Generation 3, we get 14 out of, well, we get the exact same numbers, so this row is a little bit dull. And in row 4, we get 15 out of 40, 13.3%, yeah, 0.375. I might have gotten that if I'd thought a little longer. 18 out of 40 is 0.45. And 7 out of 40 is 0.175. So, first thing you'll notice, the Hardy-Weinberg formula says that if you add all these up, you should get 1, and if you check these, you'll find that, yes, in every case, we get exactly 1, so that part works great. The other thing that the Hardy-Weinberg formula says is that if you're in genetic equilibrium, these numbers shouldn't change. They should be steady from one generation to the next, assuming that this trait isn't a selection advantage and that there's random mating and there's no immigration or emigration. These numbers float a little bit, and the reason for that is simply that our population is not all that big. Right? If you take a small sample, like if you flip a coin once, you're not going to get half heads, half tails. You're going to get a number that's not 50-50, and that if you just look at that one number, it'll make you think, well, wait, this coin doesn't do heads half the time. I just tossed it once and I got tails. The mistake there is in looking at too small of a sample, and the bigger of a sample you take, if you flip a coin 50 times or 1,000 times, you should find the number of heads and tails starts getting very close indeed to 50%. And if you look at the number of children born in a country, you'll find that, yes, it is pretty much exactly half boys, half girls. And so these numbers are drifting a little. We go 0.4 up to 0.5, back down to 0.45. That's just because in a small group, luck can throw off your numbers a little bit. So we're not thrilled to see this, but it's not a surprise either. It just means even though I was doing 40 individuals per generation, even that is not enough to completely take out the effects of randomness. However, I hope you notice that these numbers are pretty steady. I mean, 0.4, 0.35, 0.375, this isn't moving all that much. This one moves a little. This one doesn't move much at all. So you can probably imagine if I had done this with 100 or 1,000 beads, these numbers would be very close to the same one generation after another. They say in B, use the class data from part 1 to determine the allele frequencies. That's not too hard either. If we're counting alleles now, let me do a color switch. Every individual in, there we go, sorry. Each of these individuals has 2 big A alleles, so we can count this as 32 of the dominant allele. These are heterozygous, so these 16 individuals would have 16 dominant alleles, like 16 dark beads, and 16 light beads. And these individuals over here would have no dominant alleles, but they would have 16 of the recessive ones. So the numbers here are 32 and 0 for this group, because they're 2 dark, no light. These are 1 dark, 1 light, and these 8 individuals are no dark, 2 light. So if we count those, let's see what we get. P and Q. For the first generation, we got 32 plus 16 is 48 dominant alleles. For the recessive group, we had 0 here, 16 here, 16 here, a total of 32 recessive alleles. Now, this shouldn't surprise us because I counted these at the beginning. You saw that there were 48 dark beads, 32 light beads, so this kind of has to happen every time, doesn't it? We can work out the P and Q from this for the number of alleles, and it should be, this would be 48 out of 80, which is 0.6. Q would be 32 out of 80, which is 0.4, and those do indeed add up to 1. Now, if I do that for every other generation, because I was working with the same set of beads every single time, these numbers are not going to change at all. I didn't add any new beads to the table. I didn't take any off. It's going to be 0.6 and 0.4 for every single generation, so this question's a little bit boring. In real life, this doesn't necessarily have to happen because people live different amounts of time. They produce different numbers of sperm cells and eggs. Some people die younger. Some people have more kids, and so you see more of their alleles in the next generation. But in this example where everybody reproduces exactly once, the allele pool is going to stay exactly the same over time, and you're just going to get 0.6, 0.4, 0.6, 0.4, 0.6, 0.4. So that one's a smidge unrealistic, but at least we can see that our Hardy-Weinberg relationship still holds up. The P and the Q add up to 1. They have to. The next part of this, analysis 2, says use the class data from part 2 where we had individuals dying and see what kind of results we get from that. So we'll take all this and I'm going to shrink it but keep it around so it's still kind of there. That's our part 1 data. Part 1. Okay, now we'll look at the part 2 data where stuff was changing, and we'll see what we find. So, first generation, I'll put my p squared plus 2pq plus q squared here because it's a good way to organize stuff. Equals, we'll see if these add up to 1 and how they behave. So, first generation, we had 15 out of 40 individuals who were homozygous dominant. 15 divided by 40 is 0.375. Then we had our hetero group, 18 out of 40, that's 0.45. Then we had our homozygous recessive group, 7 out of 40, that's 0.175. If we add those up, 0.375 plus 0.45 plus 0.175, my calculator says that adds up to 1, so that's rather nice. But then some of them die, and we go to a generation 2. Is this still going to work? Now our number of homozygous dominant is 18 out of 33, which according to my calculator is 0.545454545. I don't have room for all that. I'm just going to put 0.545. Will I regret doing that? Let's find out. Heterozygous here, we have 12 out of 33. Oops. 12 divided by 33 is 0.3636363. I'm going to round that to 0.364. And for our q squared, the recessives of generation 2, we get 3 out of 33, which is 0.0909009. If I do that to 3 decimal places, I get 0.091. Now, if we add those up, 0.545 plus 0.364 plus 0.091, it does come out exactly at 1. Now, I suspect I was a little lucky here with how I rounded. I would not have been surprised to add these up and get like 0.999 or 1.001. Sometimes when you're rounding decimals like this, that happens and you will not hit 1 exactly. And when that happens, it basically means you didn't carry enough decimal places. And if you had, these numbers would have added up to 1. But if you total this up and you get something that's very close to 1 but not exactly, don't panic. it probably just means that rounding hurt you a little bit. It doesn't mean that genetics is broken or that your procedures were all wrong, just that you got some slightly unlucky numbers. But in this case, hooray, exactly 1 is nice to see. Now we have a generation that's 21 out of 30. That's going to be 0.7. And a 6 out of 30 is 0.2. And 3 out of 30 is 0.1. 7 plus 2 plus 1 is 10. Yep, that adds up to 1 rather nicely. And now we have our last generation. 21 out of 27 is 7 out of 9 is 0.77777, which I'll round to 0.778. 6 out of 27 is 0.22222. And 0 is 0. If we add these up, again we get 1. So, good. The Hardy-Weinberg relationship is holding up. And in part B, they say use the class data to determine allele frequencies. We can do allele frequencies just using our own data here, so let's get that done. Remember, when we're looking at alleles, each of these individuals came from, switching colors, 2 dark blue beads, and each of the hetero individuals came from 1 dark blue and 1 clear, and each of the homozygous recessive individuals means 2 clear. So if we're counting alleles, our P total is going to be the number of dark beads. It's going to be 2 for each of these individuals, 1 for each of these individuals, and these won't count because they didn't have any dominant alleles. So 2 times 15 is 30, plus 18 is 48. Where do I put this? Yeah, I'll put them here. So this is our dark or our capital A alleles. And then we're going to have our light or our little a alleles. How many of those were there? 18 from each of these, 18 from these, and then 2 times 7 is 14. 18 plus 14 is 32. Okay. We knew this would happen. We've seen these numbers before. But now they're going to change, because remember I had the graveyard where I was putting any light alleles that died? That's going to start shifting these numbers, so we really do have to go through the other rows. Here we get 2 times 18 plus 12 is 36 plus 12 is 48 again, because remember we didn't lose any dark blue beads. But for lights, we now only have 12 plus 2 times 3 is 12 plus 6 is 18. And in generation 3, we'll have 42 plus 6 is 48 dark blues, because we never lose any of those. Dark blue is the survival allele. And for lights, we get 6 plus 2 times 3 is 6 plus 6 is 12. And finally, 42 plus 6 is 48 yet again, because we don't lose any big A alleles. And 6 plus 0 is, we're down to 6 recessive alleles. Now, those are the raw numbers. To get P and Q, I have to do a little more math. I have to get the frequencies for each of these, so bear with me while I crunch that out. Here we have a total of 80 alleles. Here we have 48 plus 18 is 66. 48 plus 12 is 60, and 48 plus 6 is 54. So when we're doing our frequencies, we would say P is 48 out of 80. That's 0.6. Q is 32 out of 80. That's 0.4. Well, that's boring. We've seen that before, but let's see if it changes as time goes on. 48 out of 66 is 0.72, or 0.73 it rounds to, actually. And 18 out of 66 is 0.2727, so 0.27. Next generation, we have 48 out of 60, which is 0.8, and 12 out of 60, which is 0.2. And in this last generation, we have 48 out of 54, which is 0.888888. I'll say it's 0.89. And then we have 6 out of 54, which is 0.11111. So, if we add those up, 0.6 and 0.4 makes 1, 73 and 27 makes 1, 0.8 and 0.2 makes 1, and 0.89 plus 0.11 makes 1. So, hooray, that part is working. In 2C, they say, what happened to the total number of alleles over the course of the investigation? Did this affect the results? Well, we were running out of alleles, right? As this thing went on, we had more and more recessive alleles in the graveyard over here that we couldn't use anymore. And so the population, as we went through the cycles, started to look more and more blue because the recessive allele was dying off. And you can see that in the numbers. Here's the number of individuals who died in each generation: 18%, 9%, 10%. Here they had a lucky generation with 0%, but it undoubtedly would have rebounded, and we would have had this number shrinking slowly away because recessive traits tend to die off. If they aren't able to be passed on and they disappear out of the gene pool every generation, this may not ever completely go away, but it will become extremely rare because it isn't a survivable trait. The Hardy-Weinberg Principle says that you can expect these numbers to remain steady as long as you're talking about a trait that doesn't affect your mating potential, doesn't affect your survival potential. We aren't following that rule with this trait. This trait kills you, and so Hardy-Weinberg says, well, you're not going to get equilibrium then. This number is going to drop away towards 0, and these other numbers will increase, the ones that allow you to survive. So remember, we should only be using this for things like brown hair versus blonde hair, or things that don't really matter to your ability to survive and have kids. As soon as you start using it for a lethal disease or something like that, you're not going to find equilibrium. You're going to find that the numbers shift over time. So that's what they're talking about in part 2 of the analysis. Part 3, explain why data from the whole class were pooled. I mentioned that in a small sample, bits of luck can affect your results, and the bigger your sample is, the more reliable and the more luck-proof your results become. When they're doing a political poll, you don't talk to one person and ask them who they're going to vote for, because you won't get a reliable result that way. The best results come from taking lots of different samples in lots of different places and then combining them at the end. That means that a little fluke somewhere is not going to throw your results off so badly. So, I mean, sometimes money is a constraint, and it means scientists can't take as big of a sample as they would like, but they're always trying to get bigger samples because that leads to better results. In analysis 4, they say, in reality, each individual contributes one allele to the next generation. Identify any false assumptions that were made when choosing alleles in this investigation. Well, we assume that everybody reproduces, and everybody reproduces exactly once, which is a bit of a stretch, but it isn't extremely far from reality. The other thing we assumed was that every individual is going to pass on both of their alleles, right? If we had an individual in one generation, and let's say their genotype was they had a dark blue bead and a clear bead. What I would do next is I would take both of these and put them back on one of the plates, and then each of these would get used to make the next generation, which meant that every individual's 2 chromosomes both got passed on. It was guaranteed they would both get passed on. That doesn't have to happen. If this is what your genotype looks like, it is totally possible that you could produce one sperm cell that has the dark blue, and then you could produce another sperm cell that also has the dark blue, and another one that still has the dark blue, and you could have several children who all get the same allele from you, and you never pass this on. There is some luck involved there, and we weren't able to reflect that in the simulation. So that's at least one way that we were cheating. There are probably others. Just because you have two alleles doesn't mean that half your kids are going to get each of these. It's a coin toss every time. You could have 10 kids in a row who all get your one chromosome, or 10 in a row who all get the other, or it could be a shuffling of the two, and that shuffling is random also. So, yeah, that's at least one of the ways that this was unrealistic. In conclusions 5, they say account for the allele and genotype frequencies observed over time in part 1. We kind of looked at this already. These are the frequencies, and we saw that they drifted a little bit because there's some luck involved here, but that really they didn't drift that much. This number stayed steady in the high 30% neighborhood. This one hovered around high 40%, and this one was in the high teens. And over time, we probably would have gotten similar numbers over and over again. Occasionally, you get a fluky generation where one of these numbers will jump, but they should settle back down. And so they say, were the conditions of the Hardy-Weinberg Principle met in this population? Well, Hardy-Weinberg says if the trait is not a survival trait, that it doesn't affect your survival or your reproductive chances, and if there's no immigration or emigration, if the trait isn't lethal or anything like that, then these numbers should stay pretty steady. So you can read that the other way and say, if these numbers are staying fairly steady, then yes, that probably means that this trait did meet the Hardy-Weinberg criteria. It wasn't affecting reproductive chances. It wasn't shifted by immigration or emigration, so yeah, these numbers probably do fit. And then in conclusion 6, they say, what about for the second group? Look at the allele and genotype frequencies. Did this meet the requirements? And here we can see p squared is climbing, 2pq is declining a little bit, q squared is dropping sharply because these individuals are dying. These numbers are definitely in flux because suddenly we've said the double recessive trait is lethal. The fact that these numbers are changing means, no, we're not meeting the Hardy-Weinberg criteria, and it's obvious why. They say right up front, if there's a trait that affects your chances of living, then you're not going to get an equilibrium out of this. This trait affects your chances of living, and so unsurprisingly, we see the numbers changing. And they'll continue to change until Q is so rare that it doesn't make much difference anymore, until the number of deaths that it's causing is so low compared to the rest of the population that it isn't a big deal anymore. Only then can you start to say that you're getting back to equilibrium case. So, I hope that all helped. It's kind of a fast overview of how a population can either not change very much if you're talking about a trait that doesn't matter, that isn't a life or death condition, and how a population can shift fairly rapidly over generations if you have an allele that is favorable or unfavorable to survival. And when we talk about a selection advantage or a trait being selected for or against, this is the kind of thing we're talking about, where if you have the wrong alleles, you end up in the graveyard.